Shy guy.
So now we're going to talk about tubular transport and start with the big picture. So we start with the proximal tubule. It reabsorbs the lion's share of a variety of substances and the fluid volume. So about 2/3 of the filtered sodium and associated anions are reabsorbed in the proximal tubule. About the same amount of water, about 2/3 or so of the filtered volume, is also reabsorbed, and the proximal tubule is always permeable to water. Also, other substances to keep in mind is 100% of the filtered glucose and amino acids are reabsorbed as well, which means that there's zero amount of glucose and amino acids appearing in the urine. Also, about 90% or so of the phosphate, 80% of the bicarbonate, and about 2/3 of the calcium and potassium. Other things that are occurring in the proximal tubule are secretion of organic anions and cations, and there are transporters that do this. For the anions, an example would be para-aminohippuric acid, and there's a variety of other anions that also get secreted, and also cations such as creatinine, which is secreted to a small degree, but still creatinine is a very useful substance to measure the GFR, ignoring that small amount that gets secreted. If we look at the loop of Henle, which is made up of a descending limb and ascending limb, we start with the thin descending limb, which reabsorbs water. It's always permeable to water, and that water reabsorption is dictated by the medullary gradient. As you go down into the medulla, the concentration in interstitium increases of osmoles, and that drags water out of the tubular fluid, helping concentrate the urine. There's no reabsorption of any ions. The thin ascending limb, which is part of the juxtamedullary nephrons only, those long looped nephrons, they reabsorb some sodium and anions, and they are also impermeable to water, which is important because as you're going up the loop of Henle, you are diluting the tubular fluid because you're pulling out osmoles and leaving water behind. The thick ascending limb is the final part of the loop of Henle. It reabsorbs sodium and associated anions as well, and it is also impermeable to water. So it is a diluting segment because you're again pulling out osmoles, leaving water behind. Another thing that the thick ascending limb does is it reabsorbs other ions like calcium, magnesium, things like potassium, and there's some sodium also that's reabsorbed, a different mechanism that we'll look at in another video. And these ions, they get reabsorbed in between the cells, and that's called paracellular transport. Also, the terminal part of the thick ascending limb is called the macula densa. These are the same cells, but they're specialized to sense the tubular fluid composition of sodium and chloride. And based on what they are sensing, they send different signals to the afferent arteriole. Remember, the macula densa is right up against the afferent arteriole, so those signals are released in a paracrine fashion. And what they can do, depending on the signal, can either constrict the afferent arteriole. The signal there is adenosine, or prostaglandins can be released, which cause dilation, and that helps regulate the renal plasma flow and the glomerular capillary pressure, which affects the GFR. So this mechanism of signaling from the macula densa to the afferent arteriole constricting or dilating is called tubuloglomerular feedback, or TGF. And the same signals also regulate renin increasing or decreasing. The adenosine decreases renin, and the prostaglandins increase renin. So it's a very important sensing mechanism and signaling that has important effects on not only the GFR but also renin secretion. Finally, we look at the distal nephron, which we can start with the distal tubule. It reabsorbs sodium and associated anions, and it's also always impermeable to water, like the ascending part of the loop of Henle. So the distal tubule is also considered a diluting segment because you're pulling out osmoles and leaving water behind. There's also some reabsorption of calcium that's regulated by the hormone parathyroid hormone, PTH, that is very important in calcium homeostasis. Finally, the collecting duct, if we just group all the different cell types together and simplify things, we can say we're reabsorbing sodium and associated anions, and there's some cells in the collecting duct that can secrete potassium and hydrogen ions, and this process of reabsorbing sodium and secreting potassium and hydrogen is under the regulation of aldosterone. The reabsorption of water, which depends on antidiuretic hormone, that's a variable reabsorption capability of water. If ADH is not present, then the water channels that ADH regulates won't be inserted, and the permeability of water will be decreased, or in some cases without any ADH, the permeability to water will be zero. So unlike the proximal tubule and the descending thin limb that's always permeable to water, the collecting duct is variably permeable to water depending on the amount of ADH that's present.